Okay, so I'm going to talk about how TGF-β signaling works. So TGF-β is a dimer, shown here in this image. So it has two parts. They're linked together by this red disulfide bond. And so one of the dimers is light green, the other one is dark green. So this protein TGF-β, which stands for transformational growth factor beta, is present in extracellular fluids or media. Now, the way that this signaling compound works is by binding to receptors in the cell membrane. So if it binds to this complex called receptor 3, receptor 3 will then present the TGF-β to receptor 2. And in other types of cells, TGF-β directly binds to receptor 2. And once this binding occurs, R2 will then phosphorylate R3, as in here. And then R2 then becomes a kinase. And so inside the cell, there's compounds called Smad 2 and 3, which have this general shape with a nuclear localization signal bound through a linkage to another domain called MH2. And what happens is once you have the activated R2 complex, it will phosphorylate the MH2. So you can see now you have that yellow phosphate group, and that causes the dissociation between these two domains. And now with this type of structure, it could then bind to a co-SMAD called SMAD4, shown here in red, and also to a important beta compound. And so two of these SMADs 2 and 3 together with the SMAD4 form this trimeric structure, and the important beta will then also be added to the nuclear localization signal domain of the two SMAD 2 and 3. This then allows this trimeric complex to go into the nucleus, where a RAN GTP activated protein will remove the important beta signal, and together with various transcription enzymes such as TFE3, this trimeric complex will be able to bind to the particular segment of DNA that it's for which it is complementary, and initiate transcription of the adjacent gene. Once this is done, a phosphatase will remove the phosphates from the SMAD proteins, which will then allow it to assume its original shape, where it could then be transported back to the cytosol for another signaling pathway. So proof that SMAD3 binds only when it is adjacent to another master factor enzyme such as this OCT4, which is a master transcription factor, which is found in undifferentiated embryonic stem cells. So you can see here from this y-axis would be here DNA base pairs, and you can see that SMAD3 is in most of the sites where OCT4 is located, and so it shows that these two are complementary and they have to be present for the genes to be transcribed. So any particular gene in the cell of the same organism may have several different binding sites, but in different cell types, different binding sites will be activated. For example, in a embryonic stem cell, you can see that the SMAD3/4 complex will bind to this left binding site with the OCT4, whereas in a muscle cell, the SMAD3/4 complex will bind in this right binding site adjacent to this other master transcription factor, MyoD. And so it shows that the SMAD complex is active in many types of cells, and it depends what cell type that cell is. That will determine which binding site the SMAD complex will bind to in the gene.